Ladies and gentlemen, good afternoon, and welcome to the Shaw Lectures. Today, we are able to gather here because we are in a time and in a place where our beliefs can be pursued openly and limitlessly, and where the inquiring mind will always be encouraged and appreciated. At this event, we wish to pay tribute to one of these great inquiring minds through a tradition that honors not only intellect, but persistence and vision. My name is Paul Kolyanen. I'm a medical student here at the University of Hong Kong, and it is a great honor to have with us the Shaw Laureate in Life Science and Medicine of 2005, Professor Sir Michael Berridge. May I now first invite the Vice Chancellor of the University of Hong Kong, Professor Ip3 to give us the welcoming address.
Good afternoon, Sir Michael, distinguished guests, colleagues, students, and ladies and gentlemen. On behalf of the University of Hong Kong, I would like to extend a warm welcome you all to this Shaw Prize Lecture in Life Science and Medicine of 2005 this afternoon. The Shaw Prize was established under the auspices of Sir Run Shaw to honor individuals who have achieved distinguished breakthroughs in academic and scientific research or application, and whose work has resulted in a positive and profound impact on mankind. I would like to take this opportunity, of course, to pay special tribute to the vision of Sir Run in establishing such a meaningful prize. Sir Run and the Shaw Prize Council have selected 3 fields in which the prizes are to be awarded: astronomy, life science and medicine, and mathematical sciences. So Michael Berridge is, of course, the winner of this year's Shaw Prize in Life Science and Medicine. As part of the proceedings, the Shaw Laureate has to agree to do 2 things. First is agree to come to receive to attend the award ceremony to collect the award. And second, to deliver a lecture after receiving the award. I'm delighted that Sir Michael has agreed to present his lecture on calcium signaling in health and disease here at Hong Kong U. Like in the airline commercials, I know that you have many choices to pick. Thank you very much for choosing Hong Kong U. Professor T.M. Wong will be making a proper introduction of Sir Michael in a minute, but let me say that it is indeed a great privilege for the university to be host for this Shaw Lecture in Life Science and Medicine again this year. Once again, very warm welcome to Sir Michael, and my sincere thanks to the Shaw Prize Foundation and all the others who have made these events possible. Like you, I'm excited. I'm looking forward to Sir Michael's lecture. Thank you.
Now, may I please invite Professor T.M. Wong, the head of physiology, to introduce Sir Michael Berridge.
Dear Sir Michael, honorable guests, the VC, colleagues, ladies and gentlemen. It's my privilege to introduce to you the speaker of this lecture, Professor Sir Michael Berridge, the Shaw Prize winner in Life Science and Medicine this year. Sir Michael is well known to scientists in the biomedical field, and actually needs very little introduction. Sir Michael was born and brought up in southern Rhodesia, now known as Zimbabwe. As a young boy, he was excited by the African bush with its animals and birds, a passion that has fascinated him ever since. This passion for natural wildlife made him dream of becoming a game warden in one of the national parks. Fortunately for us, you did not become one of the game wardens. Otherwise, we don't have you as Shaw Prize this year or a speaker in this lecture. This passion also stimulated his interest in ecology when he studied in the University of Rhodesia and Nyasaland. However, he was fascinated by physiology lectures on cardiac functions. This prompted him to pursue his postgrad studies in physiology at Cambridge University using insects as experimental animal models. He studied hormonal secretion, hormonal control on secretion in excretory system and salivary gland in Cambridge and later in the United States. This laid the foundation for his subsequent research leading to his world-renowned discovery. inspired by the discovery of cyclin B as second messenger by Sutherland and Raw. And having returned to Cambridge, Sir Michael began to look for the unknown second messenger that triggers the release of calcium from its intracellular store. He made thorough literature search, acquired the necessary techniques and methods, collaborated with the right people, and transformed himself into a biochemist. Sir Michael's effort proved fruitful when he finally demonstrated convincingly. Can I have the next slide, please? He finally demonstrated that upon receptor stimulation, Ip3 is released. The Ip3 stimulate release of calcium from its intracellular store. Now, this work was published in Nature in 1983. In the subsequent year, he demonstrated the same phenomenon in other many cell types in collaboration with other laboratories. The Ip3 and calcium pathway is a very important signaling system in the regulation of cellular function. The significance of this discovery of this calcium-mediated signaling pathway is best described by the Shaw Prize in Life Science and Medicine Committee as one of the most important pathways in biology and have changed forever the way we think about prevention and treatment of disease. His achievement has earned him numerous prestigious honors, including Lasker Prize, Fellow of Trinity College, Fellow of the Royal Society, and Foreign Associate of the National Academy of Sciences, in addition to the Shaw Prize in Life Science and Medicine this year. Today, Sir Michael is going to tell us some of his more recent research on calcium signaling in health and disease. Without further ado, I call upon him. Sir Michael, please.
Vice-Chancellor, Professor Wong, thank you for those kind introductory comments. It's been a great honor to be in Hong Kong this last week. And of course, on Friday was the award ceremony, and it was a truly exciting procedure. And since I'm going to be going straight from here to get on a plane, I've been carrying my medal and my award with me so it doesn't get lost. So I thought it'd be quite nice to put it up here, and if anybody would like to come and have a look at Sir Run Run laid out in gold, please come and have a look at it afterwards. So today what I thought I would do is to talk more generally about calcium signaling. And I'm going to just try and give an idea of some of the main aspects of calcium signaling, and then also, towards the end, discuss how changes in calcium signaling can lead to some severe diseases in man. So you're probably fairly familiar with the basic idea of calcium signaling. And is somebody going to be pointing up there? You're going to be pointing, okay. Yeah. This is a double act. Professor Wong is going to follow the pointer, and so bear with him. So here we have this external stimulus acting on the cell. And in response to that, the calcium concentration changes from this very low concentration of about 100 nanomolar in the resting cell, and it goes up about 5-10-fold. And this increase in calcium can activate a large number of cellular processes. Now, this is an extraordinary versatile signaling system. And one aspect of this versatility is the fact that the calcium signal can operate over a very wide time domain. It can operate in microseconds to drive exocytosis, milliseconds to drive contraction. And then when you come down to gene transcription, fertilization, proliferation, and so on, the calcium operates over a much longer time frame. So one thing I'd like to emphasize is this versatility of the signaling system. One of the difficulties with understanding calcium is that calcium originates from two separate sources. It can come in from channels on the plasma membrane, or the stimulus can act on receptors on the cell surface. And Professor Wong also first indicated in his introductory comments how this receptor then generates second messengers such as cyclic AMP, which then releases internal calcium. And it's this aspect here which I was particularly interested in way back in the 1980s. So this now is, if you like, the way we think IP3 is being generated. It's part of a phospholipid, phosphatidyl inositol. This it has these typical fatty acid chains in the membrane, and then a phosphate, and then this inositol head group. And then there are kinases which first add a phosphate to this 4 position to make PIP, and then there's a 5 kinase which adds phosphate to this 5 position to make this PIP3. This is the lipid precursor that is used by the hormone. So the hormone binds to its receptor, it activates this enzyme phospholipase C, and this breaks the molecule at this point to liberate the lipid part, the diacylglycerol, which then activates PKC and protein phosphorylation. And this was the pathway that Yasutomi Nishizuka discovered about the same time we discovered IP3. And some of you may know that Professor Nishizuka died last year, and this was a great sadness for many of us in the field. The water-soluble product is IP3, which is then released to the cytoplasm to activate calcium signaling. Now, of course, this isn't all that there is to calcium signaling. Calcium signaling is a lot more complicated. So what I'm going to do in this lecture is first of all describe in more detail the calcium signaling toolkit. Then I'm going to say something about spatial and temporal aspects. Then I want to talk about calcium operating in development, in proliferation, and also in processes of fertilization. And then at the end, I'm going to talk about its role in various diseases, and I'm picking out manic depressive illness and heart disease. Okay, so this is the toolkit for calcium. I've just talked to you about the stimulus acting on a receptor, PLC to IP3 to an internal release channel. But we now know that cells also have ryanodine receptors to release calcium, and they have lots of different channels in the membrane to bring calcium into the cell. So these are all the on mechanisms which generate a calcium signal in the cell. But then there are the off reactions. So once we've got a calcium signal, we've got to pump the calcium out. So some of it comes back into the store, and lots of it gets pumped out of the cell through pumps and exchanges. The calcium, whenever there's a calcium signal inside the cell, some of it goes into the mitochondrion. A little pulse goes into the mitochondrion. And then during the recovery, that calcium comes out again. And this is very important because when the calcium is in the mitochondria, it activates metabolism. So it's saying to the cell, hey guys, we're going to have to start doing something like contracting or secreting. We're going to need lots of energy. So it makes sense for some of the calcium to go into the mitochondria to activate it. So that couples metabolism with downstream effector processes. So this is the total complement. These are all the components that are in the cell involved in generating calcium signals. Now, during development, what specific cells do is that they select out those parts of the toolkit which they need for their particular function. So this is a cardiac-specific calcium signal zone. So when the cardiac cell develops, it selects out those receptors like the endothelin receptor or the beta-adrenergic receptor that it will need. They select out transducers like phospholipase C or adenylate cyclase. They select out some channels, some pumps, some buffers, some effectors. And these are quite specific for the cardiac cell. And this assembly of components then delivers a particular kind of calcium signal. So that's the normal situation. I'm just going to begin now by saying a little bit about disease. So what I've talked about here now is a cell-specific signalsome, like that cardiac signalsome, which is producing a specific kind of calcium signal, and this then gives you a normal cellular response mechanism. Now, there are two ways in which this signaling system can be altered. One is through genotypic modifications. And what happens here is there's a mutation in one of the components of the signaling system. And that mutation then gives a remodeled signalsome, which now delivers different calcium signals, either signals that are too large or signals that are very small. We now know a large number of genetic diseases that are related to mutations in calcium signaling. I don't really have time to go through these in great detail. I just mentioned malignant hypothermia, which is a mutation in the ryanodine receptor. There's hemiplegic migraine, which is a mutation in one of the voltage-operated calcium channels. There's polycystic kidney disease, Brody's disease, Haley-Haley's disease, and the list goes on. But in many ways, these genetic diseases are really insignificant compared to the phenotypic diseases. In this case, phenotypic modifications where we change the expression of the signaling system. And here we see some of the really serious diseases of mankind, the cardiac hypertrophy, hypertension, a manic depressive illness, perhaps also Alzheimer's disease. These aren't due to specific mutations. They're due to the fact that the components, the signaling components in the cell have been altered. And as I say, I'll come back to this later in my lecture. Okay, now I want to talk a little bit about spatial and temporal aspects. Because to understand calcium signaling, you really have to understand how it's organized in time and in space. So almost every cell, when it generates a calcium signal, it produces a calcium spike. So it's very much a digital signal. So you have a rapid on reaction, that was when all the calcium was coming into the cell, and then you have those off reactions which takes the calcium away. So this is a unitary spike. Now in contractile systems like the heart or in the brain, these calcium spikes are on demand. So in the heart you have to have an action potential. Each time you want a calcium signal, you've got to have an action potential. So I like to think of these as calcium signals on demand. But there are many other cell types where the stimulus is there for a long time, like a hormone. So if you put a hormone on a liver cell, what you find is it starts to generate calcium spikes. But what we found very early on when we were working on the insect salivary gland was that the frequency of these spikes depended upon the strength of the external stimulus. So if you have a certain level of stimulus, you have this frequency. If you increase the concentration of the hormone, then the oscillator speeds up. So what cells do is that they use a frequency-modulated way of encoding information rather than amplitude modulation. And you all know that when you tune your radio, you usually like to have it in FM because the reception is a lot better. Now, cells figured this out a long time ago by themselves. So they use FM rather than AM. There are other ways in which they encode information. Here I've just mentioned changes in frequency. But they can also use changes in amplitude, and they can use changes in the width of the transient. And all of these subtle changes in the oscillation can be decoded by the cell. Now, I don't have time to go into this in great detail, mainly because we don't really understand the decoding in any sort of detail yet. But there are two main ways in which you can decode the calcium signal. One is through digital tracking. So every time there's a calcium spike, you get a corresponding response in the cell. So this is just like a heart cell. You get a calcium spike, you get a contraction. But then there's something which I refer to as integrative tracking. And this might be related to how gene transcription might be regulated. Some of the transcriptional factors that are regulated by calcium might use digital tracking. So when there's a spike, you get a small change in the transcriptional factor distribution, for example. But you need a number of spikes in order to move up to some new level of activation. So that's a decoding mechanism. Right, now I'd like to turn to spatial aspects. One of the important things about calcium signaling is that the calcium signal is often very precisely localized inside the cell. And these small pulses of calcium we refer to as elementary events. So when calcium comes in across the channel in the plasma membrane, our imaging systems are now sufficiently sensitive that we can see these small unitary events. And this is being called a calcium sparklet. The ryanodine receptors, when they release their calcium from the endoplasmic reticulum, this has been referred to as a calcium spark, just a little brief pulse of calcium. And if it comes from an Ip3 receptor, this is a calcium puff. While I'm on this slide, I'd just like to mention that these internal receptors are regulated by many factors. Not only is Ip3 important, but so is calcium. And so is calcium within the lumen of the endoplasmic reticulum. So these receptors are integrating lots of bits of information. Ip3, cytosolic calcium, luminal calcium. And depending on these, they will either open or not. And this is actually a very interesting mechanism because it's used in the brain, as I'll describe a little later, as a coincident detector. It can use Ip3 coming from one signaling pathway, calcium coming in from another neural input, and it can integrate these two. And that's what learning and memory is all about. It's coincident detecting. And it turns out that Ip3 might be a very important coincident detector. The ryanodine receptor is also under multiple control. It's regulated by calcium in the cytoplasm, it's regulated by luminal calcium, but it's also regulated by cyclic ADP ribose. Now, some of you may have been at some lectures of Hong Chong Lee, who was visiting here last week, and he discovered this very important second messenger involved in the regulation of the ryanodine receptor, like Ip3 regulates the Ip3 receptor. But the ryanodine receptor is also regulated by ryanodine and caffeine. Now, what I'd like to do is just show you a brief video. So this is an atrial cell from the heart, which has been activated by endothelin. And the endothelin is generating Ip3. And what you'll see in this video is bright flashes all around the periphery. And these are little puffs or sparks. We're not even sure exactly what they are yet. But these are examples of those elementary events. And then occasionally you'll see waves sweeping in from either side. So these are these elementary events. Now here's some waves coming in from either side. And notice when a wave goes through, there's a period of quiescence. It's inactivated all the channels. But then they build up again. They start fluttering about, and then you get another wave going through. So I can watch this all day long because it's just fantastic. It summarizes everything you need to know about calcium almost in one video. That it depends on local events and also on global calcium waves. And I've tried to summarize that on this slide. So here we have the elementary events, the sparks and puffs, little localized pulses of calcium. But then sometimes you can have global effects, like those waves you see creeping across the cell. And in this case, what's happening is when one ryanodine receptor releases its calcium, the calcium diffuses to its neighbor to activate that cell, and so you set up a wave which sweeps through the cell. And in this case, these are the global events. Now, some cellular processes use elementary events, and I'm going to be talking a little later about synaptic plasticity in the brain, which is using this, and contraction of ventricular cells use elementary events. Fertilization, gene transcription, proliferation, they use global calcium signaling. Okay, now what I'd like to do is to turn to calcium control of development and cell proliferation. And what's intriguing, I find, about the calcium signaling system is that it begins right at fertilization. We all began our lives with pulses of calcium. So each time a mammalian egg is fertilized, you generate a pulse of calcium. And these go on for a couple of hours, and that's what activates the developmental process. So we have calcium operating very early in development. And then, of course, we have the cell cycle, where you generate a large number of cells during the development, and then at a certain point you have a process of differentiation, and you get two groups of cells. Some where there's permanent arrest, such as neurons and muscle, and then another group of cells where there's temporary arrest, the lymphocytes, fibroblasts, salivary glands, smooth muscle. And these cells retain the option of returning to the cell cycle when given growth factors. And of course, at the end, we have cell death and apoptosis. And it's intriguing that calcium signaling, in effect, is operating throughout this whole process. So I mentioned about fertilization. So this is work from Miyazaki. And what we see here is the calcium transients in a hamster egg which has undergone fertilization at this point. And what you see here is what I was trying to talk about earlier. Digital calcium signals which is initiating development. And each of these signals has a very precise spatial organization. So here we see the sperm. This is the point of fertilization, and that's where the calcium signal begins. And now you can see with time the wave sweeping through the egg. And you can see just the beginning of the recovery, and that would be up here. Then the calcium comes down, and then for several minutes, completely quiet. Then all of a sudden, another great pulse of calcium. And if you don't have these pulses of calcium for about 2 hours, no development, no initiation of the developmental process. For a long time, it was unknown about how the sperm activated the egg. It was one of the great mysteries. But a few years ago, it was discovered that what the sperm does is actually to inject a unique form of phospholipase C, PLC zeta. And this is injected into the egg. And this PLC zeta then starts to hydrolyze the phospholipid and generates Ip3. And it's this Ip3 which then establishes those calcium oscillations you saw in the previous slide. And this then activates the oocyte. Now, I want to just say a little bit about some work which is being carried out probably at this moment. Is that right, Andy? This is work in Andy Miller's lab, and Sarah Webb is also here. Those of you who don't know, could you just stand up, because people should see who these folks are. Go on. This is Andy and Sarah. They're from the Hong Kong University of Science and Technology. And what they do is they inject ichor into the egg of the zebrafish. And the zebrafish, being transparent, enables them to actually watch calcium signaling during the course of development. Now, unfortunately, I don't really have time to talk about this in any detail, but now you know who they are. They're at the Hong Kong University of Science and Technology. You can pop along and see them. So I like this recording here because this sort of summarizes the activity of calcium within the developing embryo. And you can see an extraordinary number of calcium spikes that are going on during this phase. And again, we have this kind of recurring theme of the temporal aspect and the spatial aspect. So if you look at any one of these points, this is a point about 75% epiboly during gastrulation. There's this extraordinary intercellular calcium wave. So it begins at this point over here, and now you can see, here's the time running. You can see this wave, it's beginning to spread around the whole embryo. And here it's going and dying out on the other side. So presumably this is providing some sort of information to drive the whole process of development. Last year, I'd just like to say something about proliferation. Again, it's a vast area, so I can only touch on it very briefly. That perhaps the best example where calcium plays a role in proliferation is, of course, lymphocyte activation. So when a lymphocyte comes into contact with an antigen, it acts on the T cell receptor. And here we have our old friend phospholipase C, again, hydrolyzing this lipid to make Ip3. The Ip3 acts on its receptor to release calcium into the cytoplasm. At the same time, through a process which we still don't understand, the emptying of this store sends a signal to channels in the plasma membrane to bring calcium into the cell. And this calcium is very important in driving the transcriptional processes which initiate the process of proliferation. And one of the main targets is this transcriptional factor, the nuclear factor of activated T cells, NFAT, which is inactive when it's phosphorylated in the cytoplasm. When calcium comes, it activates calmodulin and the calcineurin, which is a phosphatase. It removes this phosphate. Now the FAT can enter into the nucleus and begin to activate transcription. And these transcriptional events lead then on to proliferation. Okay, now I want to deal with the last part and begin to deal with the control of certain diseases and how when calcium goes wrong, you may end up with some of these serious problems. And just to remind you, I'm going to be talking here about how a cell-specific signalsome might be modified through some phenotypic modification, through a change in expression of some signaling components to give you these changes in the calcium signal. And I'm going to begin with manic depressive illness. Before I do that, I'd just like to mention to you how important the Ip3 receptors are, how important the ranitidine receptors are in the brain. If you look here at the distribution of the Ip3 receptor, you can find that it's located throughout the brain region. It's particularly strong in the cerebellum. The highest density of Ip3 receptors found anywhere is found in these cerebellar Purkinje neurons. It's in the hippocampus, in the cortex, and so on. And you can see very similar for the ranitidine receptor, originally discovered as a receptor in muscle cells. It's also strongly expressed throughout the brain. This is, in fact, a section through the cerebellum, and it's been stained for an antibody against the Ip3 receptor. And again, this is one of my favorite slides, because you can see with this neuron, you can actually see in the axon running down here, Ip3 receptors. An enormous amount, of course, in the cell body, and then this extraordinary, elaborate dendritic tree where information is received by the neuron is absolutely stuffed full of Ip3 receptors. Now, what might they be doing? Unfortunately, I don't have time to go into this in great detail, but the Ip3 receptors are located out in these dendritic spines. And I like to think of these dendritic spines as the computational units in the brain. This is where information is received from other neurons. Now, in the Purkinje cell, for example, I'm not sure of the exact number, but I think there are about 20,000 of these individual spines, individual computational units. The endoplasmic reticulum is a continuous network. It runs from the soma up through the dendrites, and all of these little spines get a little bit of endoplasmic reticulum. And the Ip3 receptors are sitting in this region. Now, during synaptic plasticity, these Purkinje neurons are the neurons which you use when you learn to ride a bike or play a piano. It's these cells which you are training. This is responsible for motor activity. And what happens is you have to have a coincidence between the parallel fibers which innervate the spines and the climbing fibers which innervate the dendrites. So how does a spine over here know that it's got to modify its activity? So one of the ideas is when the climbing fiber is activated, it generates a calcium signal. And we know that. If you stimulate a climbing fiber, you get a calcium signal throughout the whole dendritic tree. So here's the calcium coming along here. And of course, as I mentioned to you earlier, calcium is one of the signals the Ip3 needs. But it also needs Ip3. The parallel fiber activates receptors which generate Ip3. So if a climbing fiber has given you some calcium, and then this fiber over here gives you some Ip3, now you've got coincidence. So if this occurs at the same moment in time, then you get a great, huge release of calcium into the spine. And this then modifies this receptor, and you have synaptic plasticity, you have some learning taking place. So that was a very quick description of how IP3 receptors might be operating in the brain. Now, I just want to mention now how we think this system is altered in manic depressive illness. And I've taken this from the Lithium Club charity, and I'm sure there's no need to read through this. You can just have a look yourself. But I think depression is a major disease. For example, in the UK, we have over 2 million people diagnosed with depression every year. And a large number of those suffer from the rather severe and extreme mood swings of manic depressive illness. Now, lithium has been used extensively for controlling manic depressive illness, and we don't really know how lithium works. The sort of ideas we've been playing around with is here we have our normal neural IP3 signal zone. So within neurons within the brain, we have our IP3 calcium cysteine operating. This gives normal metabotropic receptor signaling, normal neural signaling. Now, what we think happens during manic depressive illness is that for some reason, this system has been remodeled. And when it's been remodeled, it gives excessive calcium signals. So you get too much IP3, too much calcium. And we believe that this is what causes manic depressive illness. Now, what lithium appears to be doing, and I've included here valproate, because valproate has now been used as a very effective drug against manic depressive illness. These two drugs somehow correct this remodeled signal zone, and they bring it back close to the normal signaling system. Now, how do they do that? Well, this is a rather complicated slide, but all I want to try and point out to you is here's the inositol lipid signaling system I talked about here earlier. Here are our 3 lipids. Here's the neurotransmitter generating IP3. And then we know that IP3 is metabolized to IP2, IP1, inositol, and then it gets converted back to the lipid. So there's a constant turnover here of the head group. What we think happens in manic depressive illness, for some reason, this signaling system is set too high. Either there's too much lipid or there's too much hydrolysis. Something's gone wrong here. So you've got this turning much faster than it is under this condition. Now, what lithium and valproate do is that they put the squeeze on metabolism. Lithium blocks the final inositol monophosphatase, which takes the phosphate off the final hydroxyl group to make free inositol. Valproate blocks the de novo synthesis from glucose 6-phosphate. So in effect, by blocking the supply of inositol, the cell begins to run out of inositol. If you don't have inositol, you can't reform the lipid. So what we think happens is by limiting the inositol, you now go from this condition down to this condition. You put a little kind of baffle in the metabolic pathway. You slow down the production of inositol, and then the system comes back and starts to operate normally. I see we're running a bit long now, so is it okay just to finish this off? Yeah? Okay. So the last thing I want to talk about a little bit, because this is especially for you, this bit, so I must put it in. So I want to talk a little bit about cardiac hypertrophy and arrhythmias. Just to go back. In the case of manic depressive illness, we think that this is set too high, as I've just described. But it seems that in various forms of heart disease, the calcium signal is set too low, and this is the failing heart. So I've just shown you here an example. These are ventricular myocytes which have been taken from a failing human heart. And just this over here is the calcium transient. So this is the normal calcium transient for a normal heartbeat. And you can see a very strong on reaction. This is the calcium coming into the cell from the ranitidine receptors, and then this is the recovery process. This is the failing heart. just producing a tiny little bit of calcium, and that's why the heart just can't beat. It's beginning to fail. In addition, in the failing heart, you start to see arrhythmias. And I've got just a little video here of two ventricular cells. This cell over here is normal, and there's another cell over here which you see will be behaving abnormally. And it's showing the sort of arrhythmias that cause such a problem in the heart. So every time this goes red, these two cells will receive an electrical stimulus, and you'll see them going red as the calcium increases. This cell behaves fine for a while, and then it becomes arrhythmic. And it starts developing an extraordinarily beautiful spiral wave. You see calcium sort of whizzing around inside the cell. So if we just run it. So now they're both responding to that electrical signal, well-organized spikes. You can almost see them sort of contracting. Now here's this, it's just ignoring the signal. And it misses a beat. And now it starts responding again. And if you waited and watched it again, it would go through another period of arrhythmia. So that's one of the things we're trying to understand. To understand the heart, you have to understand the way the signal is generated. I don't have time to explain this in great detail, but I'm sure you're all familiar from anatomy classes and cytology classes that the heart has these invaginations of the plasma membrane. These are the T-tubules. They carry the electrical signal into the cell, and they activate these junctional zones to release calcium. And the calcium comes straight onto these myofibrils, and that's what gives you the contraction. Then the calcium gets pumped back into the endoplasmic reticulum here, and then it tunnels back to these regions, and you get the release. So that's going on all the time. So that's just diagrammatically. Here's the T-tubule. Here's the voltage-operated channel, puts in a bit of calcium, activates the ryanodine receptor to release its calcium. Here's the calcium activating on its receptor, and you get contraction. So that's the heart beating away quite normally. Now, one way you can get hypertrophy, which is a first step towards heart disease, is to activate some catecholamine receptors, which generate a cyclic AMP. Now, this is something that Professor Wong and his colleagues have worked on, and I thought it would be nice just to show you one slide from a very recent paper of theirs. And here are the calcium transients. This is the normal heart giving these nice little beats. And then in response to isoproterenol, which is going to be activating those catecholamine receptors, now you can see these very large transients. So you needn't have to look here. You know this data so well, you could even explain it better than I could, I'm sure. And so this is the positive inotropic response. And it's quite interesting because in the overrectomized animal, you can see, this is the overrectomized animal here, that this is enhanced enormously. And I think this is a very interesting point because I'd be fascinated to know whether in these overrectomized animals the hypertrophic response develops quicker. We could discuss that afterwards. So these are the kinds of changes in the calcium spikes which lead to the hypertrophy, which then lead to heart disease. And the way in which they lead to hypertrophy is illustrated here. And I apologize, this is towards one of my last slides. Calcium signaling is complicated. So just to orientate you again, this is the normal heartbeat. You have this opens, that opens, you get calcium, you get contraction. Now in the normal heart, there's no gene transcription. But if you increase the magnitude of this signal, so this in this case is amplitude modulation, if you increase the size of this stimulus, then you begin to start to drive gene transcription. And this increase in gene transcription is very interesting because the heart begins to express fetal genes. These genes were switched off when the animal became adult. But during hypertrophy, if there's too much calcium, they start expressing fetal genes. And this gives phenotypic remodeling. And it's that remodeling which gives you the hypertrophy, the arrhythmias, the heart failure, and then the sudden heart death. So this is to go back to the encoding and decoding of information I mentioned a little earlier. Here's the heart just giving its nice contractions, regular contractions, phenotypic stability, not much gene transcription, everything happy. If you add a hypertrophic stimulus, if you have high blood pressure, if you have too many of the drugs like isoproterenol stimulating the heart to increase its contractions, here we see now that increase in contraction. And of course, the heart beats stronger, that's what we're trying to do. But now what happens is, through some kind of integrative tracking, you start switching on genetic events. And it's this remodeling of the heart which then brings about the hypertrophy, the arrhythmias, and many serious consequences. So in conclusion, what I hope I've done is to give you just some insight in how we think about calcium, this extraordinary signal which has so many functions within the cell. And as I mentioned earlier, it's really important to understand the temporal problems, the arrangement of the spikes, the nature of the spikes, how frequently they're delivered. We also need to know how they're organized in the cell, their location, whether they are waves, whether they are little localized pulses. And then finally, if we really want to understand calcium signaling, we have to put it into the context of disease. And I think there's more and more evidence that changes in the setup, in the remodeling of these calcium signaling systems may play a very important role in the onset of certain diseases. Thank you.
Thank you, Sir Michael. Exciting lecture, starting from your very brief introduction to the discovery of the Ip3 calcium signaling pathway, to the more recent work on calcium. I think Sir Michael is willing to answer some questions. Do you have any questions from the floor?
So Michael, you gave an example of how some treatment can be administered to alter this malfunctioning, for example, in the case of manic depression. I wonder if the treatment produces permanent cure, or would the treatment have to be continued indefinitely?
Yeah, well, that's a very interesting question, and it's quite clear that the treatment has to go on indefinitely. And that's one of the biggest problems. I'm not a clinician or anything, but from what I understand, one of the problems is that when people go on to lithium or valproate, they suddenly start feeling good, and they start behaving normally, and they think they're cured, they think they're better, and they come off their treatment. But of course, the remodeled system has just been suppressed. It's not altering the excessive production of these genes. It's still remodeled. But by altering the inositol flux, you can get an adjustment. And they get ill again, so they have to come back on a treatment. But lithium is one of the most remarkable drugs because it's a homeostatic drug. And it works, it blocks those inositol phosphates through a mechanism which is known as uncompetitive. Now we know about competitive inhibition, and we know about non-competitive inhibition. But when I started reading about this was the first time I came across uncompetitive. And uncompetitive inhibition is where there is no site on the enzyme for the inhibitor until the enzyme binds to its substrate. As soon as it binds its substrate, then there's a lithium site, and the lithium comes on. Now what this means is that if the cycle is turning normally, there's very little inositol phosphates being made. Then you put lithium on, it's very few enzyme substrate complexes are formed, and lithium has almost no effect. But when the cycle is working fast, now there are lots of inositol phosphate enzyme complexes formed, now lithium can block. And the more abnormal the system, the better the lithium block is. But as soon as it gets the system back operating normally, lithium has no effect. It's almost like it withdraws itself. until it starts working fast again. And people say, well, you know, we need inositol phosphates for our smooth muscle, and why doesn't it affect all the other systems in the body? And the other neat thing is about lithium is the blood-brain barrier is impermeable to inositol. So we all get inositol in our diet. There's a high level of inositol in the plasma. So all these tissues in the rest of the body are protected by lithium because they can get their inositol from the blood. But the neurons that are closeted behind the blood-brain barrier, they can't get inositol. They have to recycle it. So they are uniquely sensitive to lithium, which again is a really neat trick with regard to lithium. But lithium has drawbacks, and I saw you nodding your head because you work on genetic defects. Lithium is a teratogen. But we've known for a long time that in development, if you put, say, frog eggs in lithium, you end up with frogs with two heads. It's a dorsalizing agent. And now we found that valproate, which has now been used in many cases as a replacement for lithium, but valproate was used initially for epilepsy, very good control of epilepsy. So there are a lot of women who are taking valproate to control their epilepsy, but they've had deformed babies. Now we think we know how that is happening because lithium is a teratogen, and it's working to inhibit the Ip3 system in the developing embryo. And we think that valproate is now doing the same thing. So we have an explanation of why the valproate is doing that. Sorry, that was a rather long, but I always get very excited about lithium. So.
Oh, yes, Dr. Lee.
Thank you very much for a wonderful lecture. I wonder the Ip3 receptor and renin receptor work together or they work independently?
That's a very good question. In general, they probably do work independently, but it's something we still don't have too much information on, and especially in things like smooth muscle. You always get ryanodine receptors and Ip3 receptors. And another cell we know where we get the two is in the atrial cell. I haven't had time to talk about the atrial cell. But in the atrial cell, the Ip3 receptors and the ryanodine receptors are mixed up together. And in that video you saw those flashes, I think I said, we're not sure whether these are puffs or sparks. Because they are so close to each other, they could be both. Because if the ryanodine receptor releases a bit of calcium, it will activate an Ip3 receptor next door. Or if Ip3 gives a bit of calcium, it will activate a ryanodine receptor. Yeah? So where we know that they are together, we think they can communicate.
Thank you.
Oh, yes.
Sir Michael, another well-known disease, osteoporosis, also involves calcium. Does this disease involve any calcium signaling at all?
I don't know too much about osteoporosis, but I think one must be careful because calcium has these two major roles in the body, one skeletal role and the other the signaling role. And the two are very different. So in the cases of osteoporosis, which is brought on by changes in hormonal balance, this may or may not involve Ip3, I don't know. But that's a separate issue. It's involved in the amount of calcium that's going into the bones. Now, having said that, it's known that many of the receptors on the osteoblasts and the osteoclasts and so on do work through Ip3. So indirectly, they may be regulating the resorption or the bone process. But to be honest, I don't know a lot about that area.
Thank you.
Yes. Angel.
There's one. Oh, right.
With many young students and PG students in the audience. Can I ask you how do you think the calcium story is fully understood? Or what's in the future? Are there more second messengers, more channels, more complicated pathways? I mean, how far down the road do you think we are in understanding calcium signaling?
Yeah. I think we're well down the road. Deep down, I would say we're probably 80% down that road. There are a lot of channels, as you well know, that store-operated calcium channel still kind of defies our ability to find it and characterize it. So I think with regard to the components, there may be a few more to uncover. The reason why I feel reasonably confident is that if you look at the biology of cells, and that's what I'm trying to do with my website, is to try and explain the biology of cells. Now, if you look at the function of many of the cells we know a lot about, there's not a lot of things there which you can say, I just don't know how that works. Now, a few years ago, you would say that. You'd say, well, I just don't know how an atrial cell contracts. And we didn't. But now we do, for example. In the nervous system, we have a pretty good idea how many of the neurons are responding in terms of entry and release. So I think we are close there, but there's still many things to uncover. The role of calcium and the work that you're doing on development, there's a wonderful kind of array of things there to study. And I think the role of calcium in these longer-term things like proliferation, like cellular differentiation in all these cell types, this is still a relatively open area. But I think this is more looking at how calcium is working in specific cell types. And I really stress it's so important now to start to look at specific cell types, because that's where we will find new messengers. Not by looking at clone cells. You know, we've done enough of looking at clone cells. But people go on doing this and expressing that. We must get away from that and start looking at primary cells. Find out exactly how an osteoblast works and so on. Not how a HeLa cell works or a 3T3 cell works. We don't want to, no one's interested in that, because it's not going to tell us anything about how proper cells work. So there's a lot to do, but my estimate, we're probably 80% there with regard to the toolkit. But we're a long way away with regard to really understanding in specific cell types. I don't know whether you agree with that or not. You might argue with that.
One more question, Sir Mike.
Well, Sir Michael, so you mentioned about one type of synapse that's in the central nervous system. And so you showed this specialization of the endoplasmic reticulum,
right,
going all the way from the soma, extending into the dendrite and into the spines and all that.
Yeah.
But how about other synapses? For example, the peripheral synapse. In particular, I'm thinking of the neuromuscular junction that some people here in the audience work on.
Right.
And so is there evidence that indeed such endoplasmic reticulum also underlies the synaptic specialization there?
That varies a lot from neuron to neuron, but I've just last week I was in Innsbruck at a meeting on presynaptic release and the role of presynaptic internal stores, Ip3 and ranitidine receptors. And there are certain neurons which rely heavily on release from the internal store. Not necessarily for activating the transmitter release, but for sensitizing the system. And there's examples where the release of the internal store actually changes the sensitivity of the presynaptic ending. And this, in fact, is the way nicotine probably works in the brain. Nicotine acts on presynaptic nicotinic receptors. It introduces calcium into the cell, small amounts. That calcium loads up the internal stores in the presynaptic ending, and then those stores start releasing calcium, and they actually start driving transmitter release. You don't have to activate the neuron. So there's a lot of excitement with regard to presynaptic stores, and they are there in many of these neurons. But again, one mustn't generalize One must pick out certain neurons. They have a particular function. And if it's important that they have presynaptic control of the internal store, then that's developed. Other neurons, you may not see it because that's not their function. They want to just give a quick burst. They don't want any modulation. They don't like this modulation business. They want calcium on demand, and that's the end of it. But other neurons, it may be much more subtle.
Thank you.
Professor John Wong?
So, Michael, I'm a surgeon. And I'd like to hear your hypothesis about this clinical situation. When I was an intern,
Sorry?
When I was an intern,
Right.
one of the things that we did when we do reasonably well at the exam was to do psychiatry.
Uh-huh.
And one of the things that we did as an intern in psychiatry was to take part in the therapy of depression. And at that time, they have a physical therapy called electric convulsive therapy, ECT. And they are quite effective in dealing with serious depression. Patients under general anesthesia, and you put these electrodes between the temples and give one big zap, and eventually they wake up and they get better.
Right.
It seemed to me that there might be some connection between the electrical activity passed between the brain and some of what you've been explaining in terms of the calcium channels. I wonder if you have a hypothesis as to how this might work, especially if you look at your final slides where you postulated that the overactivity of the cycle was might be responsible for depression, and how lithium could block some of that. How do you think an electrical impulse might work in that situation?
That's a tough question. I've no idea. No one else, I'm sure, has any ideas. What I would imagine is by releasing a large amount of transmitter, you will generate fairly large calcium signals in the soma of many cells. And you'll start to alter the transcriptional activity. And this will lead to a certain degree of remodeling. The problem with these diseases, I'm positive, is its phenotypic remodeling. The expression of certain signaling components have changed. So your ECT may simply be another remodeling mechanism. In this case, by dumping a whole lot of transmitter. That would be my interpretation. So one has to then try and devise experiments in order to check that. But it would be transcriptional activation of signaling components. And what's interesting is that so many of the key transcriptional systems are regulated by calcium. NF-AT, CREB, NF-kappa B, many of these are also very sensitive to calcium. So I think cells are constantly monitoring their calcium. It's like the government when they're looking at research that's going on in their units. They like doing assessment. Yeah? They're checking all the time whether you're not spending too much money and so on. And if you do, then they give you big trouble. So what's happening in cells is when they differentiate, they have to give a certain kind of calcium signal. If they don't, information is fed back to the nucleus saying, hey, look, it's too high. And they try and adjust. And there's lots of examples now. And that's why when you use RNAi to knock out a certain enzyme, people say, oh, well, look, there's been no change. There must be compensation or redundancy. But it's actually very interesting how cells can recover from taking out a component. Because they can look in their toolkit and they can say, this guy's taken out this voltage-operated channel. But I know I've got another one over here, which is sort of works okay. So they put that in, and the neuron can go on. So I like to think of this as kind of, I'm not going to say intelligence design, because we know this is the bad route to go down. But there is an intelligence, my feeling is, that the cell knows what it's supposed to do in terms of calcium. And if this is altered in any way, it tries to make an adjustment to make sure it delivers the signal it's supposed to deliver. It's a beautiful homeostatic system.
I think we can keep you a whole day. Before we let you go, can I ask you for a favor? We have so many young students, new researchers here. Can you give them some advice before you go?
I think the main advice is don't be scared of your supervisor. That's the first thing. The second thing is have a really good working hypothesis. And by that, you need to ask your supervisor whether the project you are doing is important. And if not, why not? So work on a good project, but be sure in your head you have a working hypothesis. That is the single most important thing you can have. You've got to understand why you're doing the experiments. Not because your supervisor has told you, but because it's consistent with your working hypothesis. This leads to that question. And without it, you're in trouble.
Thank you
And
very
after
much.
I've had a glass of wine, I'll talk about fluff wiping, but that's a separate...
Thank you very much, Sir Michael. Shall we thank him again?
Thank you, Sir Michael. And if you would please stay, I would also like to invite the Vice Chancellor, Professor Ip3 to present a souvenir to you. Shall I also invite Professor T.M. Wong and Professor S.K. Lam and Professor Paul Tam to the stage to take a group photo, please? And thank you very much. The Shaw Prize lecture is now concluded.